Okay, hello everybody. Thank you very much for attending this third talk of the series of the CRNS webinar series that the Center of Cognitive Robotics and Neutral Systems is organizing for fall 2020. Today we will have Anna Alexandra Bannach Brown. She is a postdoctoral research fellow at Camarades Berlin, is a center in Berlin of the Berlin Institute of Health, and she will present. She will give us a talk on automated technologies for systematic review. She is a research fellow, and in the past and currently she is an expert in implementing and developing machine learning techniques and text mining tools to aid the evidence synthesis research processes. But she will tell us today more about such technologies. Please, Anna, go ahead.
Thank you, Mario. Thank you for inviting me to talk at the Plymouth Robotics Seminar Series. Very exciting. I am a meta-researcher at Camarades Berlin, which is based in the Quest Center in the Berlin Institute of Health, affiliated with the Charité Medical University Hospital. So a little bit about my background. I am a systematic review methodologist. I'm a methodologist by training. I started off doing a systematic review of the biomedical literature describing animal models of depression. So as any young, enthusiastic PhD student, I jumped on PubMed and found over 70,000 potentially relevant articles. If you're anything like me, that made me do this screaming emoji face, because that was definitely too many for me to be able to go through by hand, let alone to try and convince any of my colleagues to help me assess the quality and extract information from these publications. So I started broadening my horizons to think about what current tools were available to reduce some of this burden in trying to gain an overview of a literature, of a field of literature. And this is what this talk is going to be about: some of the tools that I found, some of the tools that we developed, and some of the tools that are available to help us conduct some of this methodology. So they worked, and I went on to use some of my systematic review findings to refine and inform experimental design, so help us conduct biomedical science a little bit better. And also, I'm a strong advocate for open science, open access, open data, good research data management, etc. So scientific literature is growing and growing and growing at an exponential rate still. And what we are trying to do is gain an overview of the literature, whether we're doing so at the start of our research project to see what literature is out there already, or at the end of a research project when we're trying to contextualize the findings from our experiment in the context of the wider scientific literature. So one of the ways that we can gain an overview of the literature is using systematic review. Systematic review is a specific structured process, set of tools, structured methodology that helps us identify all of the data relevant to a specific research question. It may, in some contexts, if appropriate, be followed by a meta-analysis, which is the statistical process of summarizing the estimates from those outcomes of the group of studies. But systematic reviews are very time-consuming research, and on average they take about 67 weeks to complete, at least in the medical and the biomedical space. So why perform a systematic review? If it's so time-consuming, why would you do it to begin with? It provides an overview of the available evidence, all of the available evidence, as opposed to some narrative reviews that might select some articles that, that the author believes are relevant. It can help identify gaps in our evidence, gaps in our knowledge, where future research might be important. It provides a critical appraisal of the studies that we have in our body of evidence. It can help us identify factors that influence effects, so factors that are influencing the studies and the efficacy of the experiments that we are interested in. And it can help us inform the experimental design for new studies. And in general, it can help us reduce waste for future research. So, like I said, systematic reviews take a really, really long time. Professor Lau in 2019 estimated that if your systematic review retrieves approximately 10,000 unique citations, that it can cost upwards of $300,000 to conduct. So we've got databases, like PubMed or IEE Explore, where we're interested in finding the literature that's relevant. The more and more studies that get published and indexed in these databases, when we then go to these databases to try and get an overview, there's loads and loads of studies that we find that might be potentially relevant. So if you have poor data management skills, your desktop might look something like this. And this is really important because there's a mountain of knowledge available to us, and there's mountains of knowledge in any kind of specific scientific discipline. And it's our aim in science is to get closer to understand the truth in science, to get closer to improving our understanding of scientific phenomenon. However, climbing these mountains of knowledge doesn't necessarily need to be, a trek full of a huge backpack. Sometimes we can use technology to help us get to the top and look at that beautiful scenic route, so the scenic overview. So I'm going to talk about today some of the automation tools that we currently have in our systematic review toolbox available to us in our methodology. Very briefly, the systematic review steps broadly encompass systematically searching for all of the literature that's available, screening the articles that you find against predefined inclusion and exclusion criteria as to whether they are relevant or not relevant to your research question. Can involve categorizing the papers into drug A, drug B, or whichever groups are useful for your research question. The next stage involves extracting data from the papers and the studies that you are interested in, and then analyzing the data. So some of the research tools that we're going to be talking about aim to just kind of speed this entire workflow process up. So what do we mean by automation tools? This is a kind of very, very broad umbrella concept term to really mean any computer tool or software that can help us either fully or semi-automate one or several of the tasks in the systematic review pipeline. So those could be just even one single task that's involved, or it could be platforms and software that have tools and interfaces for multiple tasks. And our overall question is, when we're developing these tools and when we're implementing these tools, is can these tools reduce the resources that it takes to conduct this research? This research is one of the highest pillars of evidence that we have, especially in biomedical research. So we really want to be able to do it, but we want to be able to do it efficiently. So here's just a slightly more detailed breakdown of each of the individual steps of a systematic review. And I'll be referring to this throughout my talk so that you'll be able to see where each of the tools kind of slots in So I'll be focusing on mainly tools that are investigating these highlighted steps, developing and designing your research question and your protocol. Luckily, we cannot automate everything yet. Still involves human involvement, of course. So briefly, I'll be talking about polyglot and deduplication algorithms that can help at the search strategy stage, systematic review platforms and custom algorithms for selecting your studies, keyword dictionaries, risk of bias dictionaries for assessing study quality and extracting data, and including some tools to help us extract information from graphs and extract statistical data. And I'll briefly touch on a tool that can actually help us write the results of our systematic review up at the end. So firstly, the first step after you've developed your protocol is to find all of the available studies that are out there on your research question. So a very key part of this is to develop a good search strategy and make sure that the search strategy is very comprehensive, so you're picking up all of the available literature, and that often involves searching multiple databases. However, if you have experience searching multiple databases, each database has a slightly different syntax. The brackets mean something else, asterisks mean something else in the different databases. Luckily, a tool has been developed to help us make sure that we don't have errors when we're translating our search strategies from one database to the other. There is a tool called the Polyglot Search Translator, built at the Institute for Evidence-Based Healthcare at Bond University. And it takes your search from PubMed or Ovid Medline and translates it into other commonly used databases, for example, Embase, Cochrane Library, PsycInfo, etc. And this is a really cool tool. How much time does it actually save you? Colleagues looked into this. Polyglot, in comparison to doing the manual search translation, was tested with 20 expert librarians helped us, and they translated 10 searches of varying complexity. And on average, using Polyglot saves about 10 minutes per database searched. And for some of the more complex searches, it can save resources up to 100 minutes. So that's really important if your search strategy encompasses maybe 3 or 4 different databases, and this can save you time. But also it helps reduce some of the syntax errors, some of the manual errors that come in when we're doing this kind of transcript work. So yeah, that's really helpful. And after we've searched several databases, we know that there's overlap in the representation of the different databases. So one single study might be indexed in multiple databases at the same time. And to save us time, we want to make sure that we only have a unique set of articles to go through and assess. So we deduplicate all of the references that we have found. We have some tools for that to help us as well. The Systematic Review Accelerator Deduplicator tool. This is a tool that has an online user interface, and it accepts a EndNote library, which is a commonly used reference manager, and it outputs you another EndNote library. So you can then use that to go on with your systematic review. So how does it work? In 4 brief steps. It does kind of simple field comparisons first. It then converts some of the page numbers and other additional kind of formatting, which is often different between databases or different between journals. It then matches on author and title, and then it uses fuzzy logic to match names. How well does this tool perform? In comparison to just using the function that is available in EndNote, It's been validated across 3 medical searches from 4 different databases, and it has a slightly higher sensitivity and specificity, which obviously means that it saves us time. We're making sure that the articles that we've got in our library at the end are less likely to be duplicates. So we're not going to have too much work for ourselves going through them. It is a free-to-use tool available at the link, and you can either use the online interface version or download the desktop version, and it has really good help materials and help guides. Then next tools for the next step in the systematic review process are study selection. And this is where we screen the articles that we found for relevance to our research question. We are interested in doing this in an unbiased way, so we usually get 2 independent researchers to make a decision as to whether the article is relevant or not relevant. And then we can either reconcile any discrepancies, or the 2 researchers can sit down and have a discussion as to why there was a disagreement. Luckily, there are some softwares to help make this easy for us, help speed up this process. These are mainly kind of web-based user interfaces that present title and abstract for us to just make a quick decision as to whether it's relevant or not relevant. And they are really user-friendly and very helpful and free to use. For larger systematic reviews where there's over 5,000 or over 10,000 studies retrieved, we also can utilize and develop machine learning algorithms, which I'll touch on a bit later on in the talk. These tools are also really useful, and these kinds of online platforms are really useful if you might have a complex search, or you're wanting to get a lot of researchers involved in your research project. So whether that be students, or you have an international collaboration and researchers from all over the world need to log in and help with this kinds of research. Surf is a really great platform for that. There's no limit on the amount of researchers that you can have working on a project, and each researcher gets a new publication to assess whether it's relevant or not relevant. They get a new publication at random. So this is really, really helpful. And I believe I've got a screenshot here. When you go in and make a free user account, you can then go and register to be a project. Oh no, sorry, the study selection thing is covering that. Underneath that, it should say register to join. And then you can, you can have an unlimited amount of researchers working on a single project at a time. And this is just a screenshot of the interface, so how the title and abstract are presented. Along the side, you can remind your collaborators what the predefined inclusion and exclusion criteria are, and they can make a decision on the right-hand side whether the article is relevant and should be included in the systematic review or excluded, or if they're not sure, they can just go on to the next one. So even though it's not using any kind of automation technologies, this kind of software has really boosted the number of people that can be involved in a systematic review at a time, and this kind of crowd approach has also made this research technique kind of reach a broader audience. You know, students can be involved in a project and contribute small amounts rather than undertaking an entire systematic review. For machine learning for citation screening, just a quick overview of the next couple of slides that I'll be talking about. I'll briefly go into how we use machine learning for systematic reviews, probably preaching to the choir here for all the amazing robotics people. Then I'll talk about how this technique has been applied for studies, randomized controls trials, then some of the custom and machine learning approaches that we've implemented, applications, performance, and how we've also used this technique to help us identify human screening errors in our research. So typically, or in previous years, we've used supervised machine learning models. That involves training or getting your researchers to label your documents, your titles and your abstracts. Then feature selection to identify features from the title and abstracts in order to train a classifier. One of the most commonly used classifiers in this classification task is support vector machine. So really simple, and then the classifier then gets applied to the new documents, the unseen documents where we are hoping to make a prediction as to whether they are included or excluded. So one of the applications of this technique is robot search, which is developed by Ian Marshall and Byron Wallace. It takes a PubMed or an M-based RISC file. It uses a support vector machine approach as well as the publication tag in PubMed to filter the studies in your library to see whether the articles are a randomized control trial or if they're not a randomized control trial. And at the end, it spits you out a RISC file where you can then use that file for later stages in your systematic review. It is free to use and available at Robot Search. It's developed in Python and the code is on GitHub, but it also has a nice user-friendly web interface for researchers that don't have programming experience. Robot Search was trained on a data set from Cochrane Crowd. This is about 22,000 documents that describe RCTs, so that they have a positive label out of 280,000 records in total. And the algorithms were validated on the Clinical Hedges data set, which is about 50,000 articles describing randomized controlled trials. And here, with this kind of task, our aim is to maximize the sensitivity, because we want to make sure that we don't miss any potentially relevant articles. We want to make sure that we are seeing all the articles that are going to be relevant to our research question, and then maximize specificity as much as possible. So removing all the kind of all the documents that are not relevant so that we can save ourselves some time. And we have also used a similar approach to develop custom algorithms for specific projects. And this is work that we've done in collaboration with the Epicenter, James Thomas, National Centre for Text Mining in Manchester in the UK, and implemented in the Systematic Review facility. Rayan, one of the platforms that I spoke about, or that I touched upon briefly, also uses a kind of similar approach. So one of the applications of this was for systematic review of animal models of depression, where we had over 70,000 potentially relevant records. We took a random subset of those and got some gold standard human labels on that. Subsetted that into a training set and a separate validation set, and then trained SVM algorithm and then applied that to identify the relevant studies out of the 70,000 records. And we were pretty happy with the performance. We had a bag of words feature selection and a linear SVM, so not very technologically advanced implementations. But they worked for us because they gave us really, really high sensitivity. We had a predefined criteria that we wanted the sensitivity to be above 95%, which is what we estimate 2 independent human researchers to be able to perform at. And then we were really happy with the specificity that we got. We then used this approach, this same machine learning algorithm approach, to help us identify human errors that we hadn't picked up on in our training set. So we took the training set and applied k-fold validation. So we took one subset of the training set away, used the rest of the labels to train the algorithm, and apply. Oh, why is that not working? Yes, to apply the algorithm's prediction onto those studies, and then took a second subset away and did the same for the entire subset. And then we ranked the articles, or ranked the records. So we had the machine assigned score and we had the human assigned score, and we could then highlight any differences where the machine thought that it should be excluded and the humans had actually included it, and where the humans had excluded it and the machine thought that it was included. So this was really helpful for us to actually identify where, despite having a gold standard training set, 2 independent reviewers with a third for any reconciliation of any differences, we still identified errors. And once we had updated the quality of our training set, we were able to get a higher performing classifier. So while we maintained the sensitivity of the classifier, we increased our specificity by about 2%, which doesn't sound like a lot, but when you have over 70,000 records, that's like, 1,500 records that you don't have to manually search through in duplicate. So after applying the algorithm, we were left with a set of 18,500 pretty likely to be relevant records, and that greatly reduced the amount of human effort and the amount of resources required to screen through and get to the next step of the systematic review process. So after we've got the articles that are relevant to our research question, we can start annotating them for things that are important to your research question, whether that be the type of study design that they have utilized, whether it be key experimental design features. Maybe it might be you're interested in the age of the participants that have been used, or the species of the animals that are used, and some of the biomedical or the types of cells that have been used. So with 18,000 documents, this is still a number to go through by hand. So we decided to develop some regular expression dictionaries. Again, a very simple technological solution, but implemented could help us quite a lot. So just for those that aren't aware, regular expressions, I like to explain this to medical people as kind of keywords on steroids. So it's accounting for different spellings, American spelling versus British spelling, accounting for capitalization, just a kind of keyword that will help you pick up the strings that you're interested in picking up. So we developed these for some key kinds of areas in biomedical research, mode of delivery of some of the interventions. So that might be whether the intervention was administered subcutaneously or intraperitoneally, whether the intervention was administered orally, etc., etc. We developed some dictionaries for the models that were used in our field and the types of drugs that they were used. So this is work that we developed in collaboration with ContentMine, which is a text mining company that's out of Cambridge University. So simply developing these regular expression dictionaries and applying them to all of our relevant records, we were able to actually get really, really good information to be able to categorize and group the documents. So this is just a quick screenshot of the depression literature tagged by the frequency of the drugs that were mentioned. The area of each of the squares is the number of documents that the term appears in. And the color or the kind of shade is proportional to the average frequency that the terms appeared in each document. So those were quite useful for us to help us gain an overview of the literature, to be able to group and categorize the documents, and to be able to extract the keywords from the documents that we were interested in, and prioritize for the next stage of data extraction. This is still under external validation, but another colleague of mine, Caitlin Hare from the University of Edinburgh, has also applied this technique in another biomedical domain, in Alzheimer's disease. And this is a screenshot of her lovely dashboard. So in Alzheimer's disease, they're interested in many kind of different types of interventions. So here there's a lovely sunburst plot that really can help you get a beautiful overview of all of the literature that is in this space. And she has also applied a technique that I'll touch on later on in the talk, about some of the reporting quality of the documents. So we've also got information from tables and graphs, which is quite frequent in the biomedical and the medical literature, that information is not just presented in the text, but also in graphical and tabular formats. So there's some lovely tools that have been adapted from WebPlot Digitizer, which have been integrated into a user interface to allow users to select and highlight graphs from publications, and simply click and select the data points in the graph within each publication, which then the numerical information from the graph then gets automatically extracted. So this saves a lot of time. The comparison to using a tool like WebPlot Digitizer in comparison to doing it manually using something like Adobe Ruler, or colleagues of mine have said back in the day they also used to print off documents and physically measure it with a ruler. I'm lucky that we have automation tools to help us for this now. On average, reduces the amount of time required for extracting information 5 minutes per graph, or almost 6 minutes per graph, which is quite significant. And it also helps us improve the accuracy. When you're measuring on a desktop ruler using Adobe, you can have errors depending on the resolution of the screen, and how accurate the reviewer is at clicking the error bars, etc. But WebPlot Digitizer really helps with that. Another tool that I want to mention is StatCheck, which is developed by Michelle Nugent in the Netherlands. And this is a tool that uses, that's looking for statistical reporting in text. So while we are interested in the raw data as well from the graphs, and we might also be interested to extract from publications what statistical tests they were doing. And this is a really neat tool. It also has a very nice user interface for you to upload your studies, and it will identify the statistical reporting in the text, and it will also actually run, because statistical tests are based on frequencies. It will run the reported values, and it will see if there's any discrepancies in the expected p-value versus the p-value that was actually reported in the article. So that's pretty neat for identifying any discrepancies or any errors in publications. So the next stage in the systematic review pipeline is to assess the study quality. We've got lots of studies in our systematic review, and we've got some really good information, but we want to understand to what extent the information that we've got is potentially at risk of bias. We want to do a critical appraisal of these studies to understand if there is any issues with the reporting, etc. So some of the tools that have been developed for this, one that I touched upon earlier is a regex dictionary for reporting of the Landis criteria, which are randomization, blinded assessment of outcome, and sample size calculation. And the development of these tools involved manually extracting phrases and information from documents where these criteria had been reported in articles, and then compiling those into a dictionary. The tool takes CSV input and outputs a kind of true-false. Some of the PDFs, well, most of the PDFs that we have available to us need to be converted to text using PDF to text converter, and there's a number of different ones that are available. And then it spits out a true-false, whether the article contains any mention of these measures to reduce the risk of bias. This tool, developed by Jeanette Bohor and colleagues in Edinburgh, was tested and validated in just under 1,000 studies on animal models of stroke. And in comparison to gold standard dual independent human reviewers, it performed quite well in randomization and blinding in terms of sensitivity, slightly lacking in specificity. Sample size calculation, we had a really kind of a tough time with, because it's not reported very often in the literature. So we need more work on this, and there is currently more work being conducted on this. Shenying Wang, a PhD student at the Camerades Edinburgh group, is looking at implementing new and improved machine learning techniques to try and both update the existing dictionaries as well as just completely have a new approach to the way that we have tackled this problem before. So this is in work that's conducted in kind of more biomedical research. There is also work in medical research that has been conducted. Ian Marshall and Byron Wallace have made a tool called Robot Reviewer that helps us assess the risk of bias of clinical trial information against the Cochrane risk of bias tool. And they've employed a kind of distance supervised approach. They linked full text articles converted from PDF. They kind of loosely linked them to data that was in the database that was in a kind of structured format. So they had unannotated PDFs and linked those to the kind of structured information that was available in the database. They then used algorithmically annotated the PDFs, or annotated the full text articles with the information that they subsequently found, employed their algorithm and got out these risk of bias assessments, as well as the supporting quotes. So the tool that they have available will highlight the text that it thinks in the article that it has. It highlights the text that it thinks is relevant to the domains of the risk of bias in the Cochrane tool. So it also is really free to use, online, nice user interface for researchers that don't have programming experience. It takes your PDFs, does all these processes in a moment's notice, and outputs the either a Word summary or an HTML summary of all of the documents that you have uploaded. And this was validated on the CDSR dataset, and they looked at trials that had been, so it was an interesting way that they validated it. They looked at trials that had been included in 2 or more systematic reviews. So then they could actually compare independent reviewer teams against each other and compare it to the tool itself. So on average, the accuracy of the robot reviewer tool was slightly under that of 2 independent human reviewer teams. But still, it's interesting to note that our kind of standards of having 95 or 99% human accuracy, and when we get separate groups of people to do the same exercise, they also don't necessarily achieve this performance. So the last step before we publish our systematic review is the meta-analysis step. And this is an optional step in the process if you have extracted quantitative and numerical values. There's a number of different tools that have been developed to enable researchers to take the information that they have extracted into a database without too much pre-processing, and actually be able to conduct their own meta-analysis in R without any programming knowledge. So this is a tool built by Shenying Wang at the Camarades group in Edinburgh. And it's, again, a very simple solution technologically, but is an incredible help to researchers trying to utilize these methodologies. So it accepts your CSV data, and it's a Shiny app that essentially conducts the meta-analysis for you by just clicking the information that you want. It'll help you even adjust the plots so that they're ready exactly for your publication. And it'll accept different formats that you're interested in, and it will accept different models and different heterogeneity estimates that are useful in the meta-analysis stage. Yeah, this was just to highlight all the different plots that it will help you do. So it will not only help you conduct the meta-analysis and forest plot, which is a reasonably standard output, it'll also do your meta-regression calculations, your subgroup heterogeneity plots, as well as your meta-analysis for identifying publication bias. So funnel plot and trim and fill and Eggers regression analysis, which is very useful. So you have got all your results, you've got all your findings. Now you just need to write them up and report them. One of the tools that's available to do this in the medical world is RevMan. And the group at the Institute for Evidence-Based Healthcare at Bond University developed a tool that will help you generate accurately reported sentences from your statistical analysis in RevMan, so that you can simply copy and paste them directly into your results section. This is quite a neat tool, especially for researchers that aren't statisticians. Not everyone is a statistician, and it helps researchers just remember everything that they need to report, everything that they need to, all the numbers, all the degrees of freedom, all the confidence intervals that they need to report in the results section of their publication. And it's still under external validation, but the feedback from users so far is very positive. So just a quick summary before I go on to talk about some of the future developments. Briefly mentioned Polyglot, the deduplication tools, custom algorithms for citation screening and systematic review platforms. I didn't mention, but the tool available also on the Systematic Review Accelerator helps you retrieve full text PDFs through EndNote. There are custom keyword dictionaries to help us annotate and extract data from publications, conduct critical appraisal of documents, extract information from graphs and statistical information, and tools to help us report our findings well. These are just some of the tools that are available. There is an entire website of systematic review tools called systematicreviewtools.com, which is a list curated by Anthea Sutton and Chris Marshall. And there is also a kind of package of tools available in R to conduct some of these, steps purely in the R environment. But the tools that I've been talking about today, and also the other tools that are available, there are so many tools currently being developed at the moment, and the tools are kind of at various stages of development. Some of the tools are purely code-based, so they're only available in Python or in R or in C#. Some of them have a user interface, and some of the tools, as you might have seen, are also developed for very, very specific use cases. So we know that they work in biomedical research, but we're not sure how well they work in other use cases, in, for example, environmental science, or if you're doing a systematic review of policies, for example. Also, if you want to use several tools in your systematic review pipeline, you will need to take your data, put it into one tool, gets processed, take it out again, maybe do some pre-processing in order to be able to import it into another tool. And given that systematic review has many different steps, this can be really time consuming, and it can be, error prone if you're doing a lot of pre-processing or a lot of conversion from one format to the other. So you might have heard about the FAIR data principles. FAIR stands for findable, accessible, interoperable, and reusable. So we also want to bring this concept to the tools and the software that we use in systematic review. Systematic reviews are tools that are used to help us improve our evidence so that we can make evidence-based decisions. We also want our tools and the software that we use to help us with this as well. So interoperability for evidence synthesis, we aim that tools are interconnected to allow for seamless exchange of data from any tool. And we want users and researchers to be able to create customizable pipelines. So you might have some tools that are, again, relevant for one use case, but a researcher from another domain might want to use them. We want these to be able to be kind of plug and play. And this is one of the principles of the international collaboration for the automation of systematic reviews. We want new tools and new development to contribute in a shared and kind of interconnected way. We don't want to, we want to make sure that the tools are immediately usable for researchers. So therefore, we have in development, we're working on an integrated interface project. And this is a kind of one of the ways that we envisage that we're going to be able to achieve this interoperability between the tools, between tools developed in different programming languages, tools at various levels of kind of development. Some might be alpha, beta, or some might be already released a long time ago. And the aim of this project is to develop a set of technical standards to improve the interoperability of our tools, and also to increase the transparency. Even if you are a systematic review researcher, going on to systematicreviewtools.com to see what tools are available for you. It's a huge, huge library. It's a difficult place to start. We also aim for the I3 project for a integrated interface. So making sure that the tools that are compliant with the technical standards, the ones that provide enough information to be able to be implemented, can be implemented on the back end, so that users are able to use them in a front-end user interface in a seamless workflow, while the back end kind of bears the grunt of that pre-processing in a way that's also reproducible. So when we're doing processing of data, if this is done by hand by a researcher in Excel, we're not sure when errors might or might not have crept in. When we do this programmatically, we can obviously go back and check and reproduce that as well. So the aim of this project is to reduce the barriers to use. So there's lots of systematic review tools, and they might or might not have a user interface. For some researchers starting a systematic review, if they don't have anyone in their research group with programming experience, they're not able to utilize any of these tools that are going to reduce the resources that they're going to be using. And also the workload associated with utilizing automation tools. So some of the tools that you might want to use that might be available in your domain might still be in alpha testing phase. And if you want to implement it, you need to learn Python and you need to be able to run it yourself. So the end goal of this project is really that there is an effective ecosystem for evidence synthesis tools as well as data, where new tools are openly available to all researchers if they would like to use them, and that tools are interoperable. And this is the same as well for systematic review data. This pipeline, this interface also allows that data can be translated and reused in a way that, for example, if there is decisions or data extracted from one record, so one bibliographic record, that other researchers who have identified that record as being useful for their review might be able to make use of that information as well. We aim for that new tools are developed effectively from user needs, and we want, what we often see is that tools get developed for one specific use case, but then because the systematic review has been finished, the tool is no longer maintained by that research group. But to do this, we require, not just researchers getting together. We also need buy-in from other key stakeholders, such as funders and educators as well. We want to make sure that the tools that get developed just for alpha or beta get to be used by the wider audience. So that's just some of the examples of how we are applying quite simple automation techniques, but I hope I've been able to display how much of a time saver, how many resources these tools are saving for this methodology, and that these tools are really helping evidence to become, research to become more evidence-based. Personally, I find it really exciting to work at this kind of intersection of fields. As a methodologist, working with machine learning experts and computer scientists and engineers, we can really learn a lot from each other and work together in a fast evolving space, so that we can harness some of the really good enhancements that are being made in the machine learning in the computer space, and we can actually harness them and use them in other research fields as well. So I just want to highlight some of the amazing developers that I have been fortunate to work with. Nico Rydell here at the Quest Center, Matt Carter, Justin and Connor from Bond University, Jing Liao, Shenying Wang and Chris Senna from the Camarade's Edinburgh group. And thank you all for your time today. And if anyone has any questions, if there's time, happy to take questions.
Thank you very much, Alexandra, for this presentation. Yes, so we have some time for questions. Are you ready? Okay. Yes, probably there might be people who must leave in a few minutes' time for other meetings.
Yeah.
However,